Welcome everyone. In this class, we are going to learn about the nomenclature, how these viruses were named, and we will study about classification also. This is what we call taxonomy. Okay. Now, these viruses are most successful parasites. Why am I saying this? I'm saying this because when this virus affect or infect an host, they first of all they will bypass their immunity. Then they safeguard themselves, and somehow if they reach their nucleic acid, they hijack its machinery. They will use their polymerases, their ribosomes in order to replicate their own DNA and also to form proteins. Okay, and in the earlier stages when viruses were recently discovered, so that time viruses were classified based on certain criterias, such as what are the hosts they are affecting, like types of infection they are causing, and what are the major causes which they are causing, and what are the vectors which bring those viruses to human. And later, when we developed certain techniques and advanced methods, we tried to classify them according to different method. So earlier various methods were used again, like what was their nucleic acid, like DNA, RNA, either it is single stranded, double stranded. Even we'll include positive sense and negative sense into it. Okay, then if the DNA segment is segmented or not, right? Like this fellow right here is a complete DNA. It contains only one segment. Okay, so this is not segmented actually. But here, this is a virus containing three different segments of DNA. So I've written here multiple segments. You will see these kind of thing in influenza viruses also. Then comes according to the size, like how big or small viruses are compared to one another. Okay, then we'll classify them according to capsids also, right? Like how is their shape, icosahedral, helical, etc., or complex. Okay, then if there is envelope present or not. Like these kind of classifications were used in earlier days in order to classify viruses. Now we will basically discuss about the nomenclature, how these viruses are named. If you discover any virus in future, I hope this will also help you. Like code for bacteriophages. Okay, we use certain codes for bacteriophages. Now bacteriophages are those viruses, these viruses which affect bacteria. Okay, they don't affect humans or animals or plants. Okay, but they will infect uh, bacterias. So for these kind of viruses, we will use codes like T1, T2, 5174, etc. And for vertebrates like us, like mammals, we use this disease like measles. It is a kind of disease. We use smallpox, FMD, foot and mouth disease. Okay, and some names are placed after the origin as well. Okay, I'm sorry for the voice issue in this earlier, but I will try to improve it. My bad. I'm really sorry. Okay. Then comes some are named after the origin of cities also, like Newcastle's disease. This Newcastle is an area, Norwalk, and Ebola is also an area. Okay. And if you talk about insect viruses, then insect viruses are named after the effect of infections, like what infections they are causing to arthropods or insects. Okay, then if you talk about plants, they have two criteria for their nomenclature. First is which host they are affecting, and second is what is the symptom. Like in potato dwarf virus, potato is its host and dwarf is its symptoms which this virus is causing, and same goes for tobacco rattle virus. Okay, now we will directly study about taxonomy. Okay, now taxonomically, when viruses were actually classified, and then various methods were used to classify them. Among them, first was what disease they are causing. Then we moved to the morphology, in which we used what is the shape of capsid, whether it is enveloped or not. It was included together in this morphology section. And then comes host organisms. What kind of host organism it was affecting. Then also according to nucleic acid, it was we tried to classify them according to what nucleic acid they have, whether they have DNA or whether they have RNA. Okay. Then came into the existence which is ICTV, International Council for Taxonomy of Viruses. Okay, and it used order, family, subfamily, and even genus also, right? But they excluded kingdom, phyla, and class. Okay, so that's how they try to classify them. And what we use nowadays is the Baltimore classification. This is the most recent classification and accepted worldwide. Okay, now it divides these viruses into 7 classes. The first class includes those viruses which have double-stranded DNA. It does not matter if it is positive sense or negative sense. The DNA will replicate first itself and make its own copies, and this DNA will form mRNA and will form protein also. Then comes second class, which is single-stranded DNA viruses. Okay, so this single-stranded DNA virus will replicate and form double-stranded DNA virus, and which will again split and form single-stranded. And this single-stranded DNA will form double-stranded and then will form mRNA. Like if it goes from this process to this process, then it can form mRNA also. Okay. Then comes class 3. Class 3 includes double-stranded RNA viruses, which will replicate and form single-stranded RNA virus, and double-stranded RNA will be formed at last, right? And this double-stranded RNA can directly form mRNA, from which it can form proteins also. Okay, and then comes class 4, which is positive sense RNA viruses. Now this positive sense RNA virus will convert themselves first into negative sense, and then they will convert themselves to positive sense again. This is how their replication work, and related to their transcription, though this positive sense will convert into negative sense will then form RNA viruses. This is its transcription, in which DNA or RNA can form mRNA, and later it will form protein from this by the process of translation. And in class 5 we contains negative sense RNA viruses. These negative sense RNA viruses will form positive sense RNA virus, then again will form negative sense RNA viruses, and these negative sense RNA viruses can directly form mRNA, then again to protein part. And we'll move to sixth class, that is retrovirus class. Now retrovirus, you must have heard about these viruses. This in, I think it was in HIV, yeah, in which single-stranded RNA, the HIV virus contains single-stranded RNA and will use retroviral enzymes to form double-stranded DNA. Then again it will form RNA, and this is how their genome will replicate. Now this single-stranded RNA will form double-stranded DNA to mRNA, which will further form proteins. Okay. And the last class is the seventh class, which is reverse transcribing DNA viruses. These are special viruses because they have double-stranded DNA but will form single-stranded RNA, then again will form double-stranded DNA to replicate themselves. Okay, now these double-stranded DNA can directly form mRNA, which will further form proteins. Okay, now this is how its replication and protein formation works. Now that's it for this class. Thank you very much.